Welcome to the Chapter 7, Section 3 podcast on cell boundaries. We're going to talk about the what goes in and what goes out of the cell. First thing we need to talk about is the cell membrane, which is the barrier around the cell. And some cells also have a cell wall, and we'll find that with plant cells, and it's a stronger, more supporting layer as opposed to the kind of thin, flexible layer of the cell membrane. So what's the function of the cell membrane? This regulates what enters and leaves the cell. It also provides protection and support. You can see I put a little border crossing there, talking about regulating what enters and leaves. Here's a picture of it, and we'll go into this more detail as we talk about this. But first thing we want to stress is it has a lipid bilayer, and here we go. Here's a lipid, which we talked about in Chapter 2. And when we say bilayer, we mean two layers. Here's one layer of lipids right here. Here's a second layer of lipids right here. And the lipid bilayer is very flexible, so that the cell can move, and it performs a barrier. And because of the non-soluble nature, like these are not water soluble parts of the lipid is not water soluble, it makes it kind of a watertight barrier here. Also inside of it are proteins, and these protein molecules are found inside the lipid layer, and there will be a carbohydrate molecule attached to them. A cell wall. The cell wall is mainly to provide support and protection for the cell. And you can see here this, I guess, pink layer right here would be the cell membrane, and these green layers right here would be the cell wall. You find them in plants, algae, fungi, and some prokaryotes, and we've seen it already in a plant cell in the Elodea. The cell wall is outside the cell membrane, and it's porous, meaning it allows in water, oxygen, carbon dioxide, and other substances to pass through. It's not as restrictive barrier. So let's talk about diffusion. And since organisms live in the liquid environment, we need to regulate that movement of particles from one side of the barrier to the other. So let's talk about concentration first. We've talked about a solution. It's a mixture of two or more substances, and we talked about the solutes being the substances dissolved in the solution. So the concentration of a solution is the mass. The concentration of a solution is the mass of solute in a given volume of solution, or in other words, mass divided by volume. Here we see how high concentration, there's a lot more mass of solute in the solution here. Here we see a low concentration, you can see the lot less molecules there. So what happens during diffusion? This is true for solutions, is that they tend, this is important to remember, tend to move from an area where there is more concentration to an area where there's less concentration. And then we call this process diffusion. So when you have enough of these particles move from through the solution, and it's equal throughout the system, that means the concentration is the same throughout the system, then you've reached what's called equilibrium. So if you look at this picture right here, there's a higher concentration of solute on one side of the membrane compared to the other, and then the molecules move from the side of the membrane with the high concentration to the side of lower concentration. They diffuse across the membrane, and then eventually equilibrium is reached. At that point, the particles do continue to go back and forth across the membrane, but they go in both directions, and that's important to remember. Once equilibrium is reached, the movement of the particles does not stop. It continues, but it's just that the particles move in either direction in equal amounts. Oh, here we go. You can see it once again. Here you have high concentration, low concentration, particles from high concentration move to the other, to lower concentration, that's diffusion, and then when we reach equilibrium, an equal number of particles travel from one side of the membrane to the other side of the membrane, maintaining that equilibrium. There is no energy used for this. It's random particle movements. That's what causes the particles to move, to diffuse across membranes. There's no it's without energy, this process. So what's osmosis? Osmosis is the diffusion of water through a selectively permeable membrane, not small particles, but water itself. Here's how it works. You have a membrane there, and the membrane in here does not allow the sugar molecules to move across it, but it will allow water to move across it. And what's going to happen is the water is going to move from the dilute concentration to the concentrated side of the water. Concentrated side. So water tends to move from highly concentrated region to a less concentrated region. And we're going to, if you look at two solutions, the more concentrated solution is called hypertonic. And here's a picture of hypertonic, and you can see that with the hypertonic, you can have it's a very you have a very turgid or very puffed out cell. Solution that's below strength is hypertonic. I got these backwards here. Hypertonic, you can see, is that you have more concentration than you do water, so your cell is going to be collapsed. Hypotonic, you have more dilute solution, so you have more water in it, you can have a more puffed up cell. And then when the concentrations are isotonic, same strength, that's this situation right here. Osmotic pressure. So osmosis exerts a pressure known as osmotic pressure on the hypertonic side of the selectively permeable membrane scale. So if you look at this, beaker right here, you can see that there's a solution here, there's more particles in here, so the pure solvent, meaning there's no particles in it, what's going to happen is the water is going to diffuse across the membrane into this side right here. And that's what we call osmotic pressure. It's going to produce a movement of water into the cell, and as a result, the cell will increase until the cell becomes full and reverse. In this case, it's going to produce a net movement of water onto this side of the tube. And this is just talking about how cells are not in danger of bursting for the most part, and cell walls also will prevent cells from expanding under tremendous osmotic pressure. We talked about support. Alright, facilitated diffusion. Cell membranes, they have the protein channels I talked about before, and these are carriers to make to allow particles to move across a membrane. And when you do this is called facilitated diffusion. And one thing you have to know about the cell is that there's going to be many different protein channels on a cell, and they're going to be different, and these channels will specifically move one particular substance across the cell. They just won't allow any substance or all substances. They're made for just one substance to move it across that membrane. So let's look at this example here. We have you can see we have the particles are being moved across the membrane from an area high concentration to an area of low concentration. And so with facilitated diffusion, we're going from a high concentration to an area of low concentration on that side. Now, active transport is when we want to move it against the concentration gradient, against the concentration difference. This is called active transport. Normally, the molecules won't want to move through the membrane they'll need to be forced through the membrane to go from an area of low concentration to an area of high concentration. This requires energy to do so. And once again, it's the same idea, as that the molecules are carried across the membrane by the proteins that we saw embedded in the bilayer. And this just talks about moving it against the concentration gradient again. So here we go. Energy. It causes the protein channel to change shape, and the molecule is moved from low concentration to high concentration. And now if we want to move large molecules, we have to use a different process, and one of the processes is moving something in is by endocytosis, taking the process of taking material into the cell by means of infolding or pockets of the cell membrane. And then there are two, and then in the end, once it gets into the cell membrane, you're forming a vacuole within the cytoplasm. Here's two examples of it: phagocytosis and pinocytosis. Let's look at phagocytosis. In this case, you have extensions of the cytoplasm surround a particle and then packaged within the food, within a vacuole. And here are those extensions right here. You can see they're like little arms reaching out to pinch this red molecule right here. And of course, once again, it requires a lot of energy. In pinocytosis, what happens is the pocket is created right here, and then when it's filled with the liquid or whatever material they want, it pinches off and forms a vacuole within the cell. Exocytosis is the opposite. It's when they want to release large amounts of material from the cell. You do it through exocytosis, and essentially what happens is the vacuole moves to the cell membrane, it fuses, and then that opens up the vacuole, and the contents then exit out of the vacuole and therefore out of the cell. On to the quiz.